Okay, so this is Dr. Granier with Bio 208. This is our Lymphatics, Chapter 22 in our Martini Nath book with Pearson. This is going over the specific immune defenses, so it is a follow-on or a second video in our series related to Chapter 22. Okay, so let me go to the shared content. All right. So right now we have finished hopefully reading chapter 22, and if you've listened to my previous videos, you know that our lymphatic system is about returning fluid to the body, and that as we return that fluid to the body that's leaked out of our capillaries through the net filtration pressure, we are going to check it and clean it and filter it. Now, for pathogens, we are going to see that they are trying to get into our body and do us harm. So our non-specific defenses, as you can see in this graph, are going to be our kind of first and second line defenses here, and we are going to see that it doesn't matter what the pathogen is, these are the ways we are going to try to prevent it from getting in. Now, once we get into the pathogen has either evaded or has propagated to a manner that it's overwhelming our non-specific defenses, we need to have a better way to target it. And so that's where our specific or acquired immunity comes into play. And the specific or the acquired immunity is going to be, again, where our T cells are going to get activated and are going to generate that cell-mediated arm of the system. And then our B cells will get sensitized and then converted into plasma cells and start upregulating and producing our large number of antibodies, and that's going to be our humoral side of the equation. And the goal is that between the cytotoxic T cells killing pathogen cells, killing cells infected by a pathogen, and the humoral side sending out, again, these little armies of proteins that can find, tag, attach, and highlight where a pathogen might try to hide, we should hopefully be able to quickly and more efficiently find and kill our pathogen. Okay? So when we look at the specific or adapted defenses, it is very heavily dependent on the use of our T cells and our B cells. All right? Now, the cytotoxic T cells, suppressor T cells, the memory T cells, that group of T cells is definitely part of the cell-mediated arm of our adaptive immunity. And the goal is that the cytotoxic T cells are looking for abnormal cells or cells displaying pathogen antigens infected by pathogens so they can target and kill it. As long as the cell is not producing antigen material, pathogen material, or not acting abnormal, the cytotoxic T cells will not be able to identify it and kill it. So that's part of the reason why for some viruses, you can have them in your system laying dormant and you don't kill those cells until, again, the cell starts to produce and actively promote the pathogen and the antigen-specific material that the pathogen has the cell create. All right, again, B cells are going to get sensitized to antigens that hopefully are from that pathogen and not from our body. And they are going to have, again, those pieces and parts of the Y part of the protein that are going to specifically be able to, like a puzzle piece, perfectly find, fit, and target and attach to that antigen that we associate with the pathogen. So they can find, again, antigens that are on our cells that are part of the cell making and kind of like hijacked by the pathogen in case of viruses and actively producing lots and lots of antigen and viral and whatever pathogen-like material. They can find pathogens that are cell-like bacteria or little protists that, again, that are displaying the antigens. Antibodies do not cross the plasma membrane. So as long, again, as a cell is not making antigens on its plasma membrane, they will not be able to find your cells that are, being over hijacked or taken over by the pathogen. All right, antibodies do not kill. So the other thing is the antibodies are just going to find and attach. They are not going to actually kill the pathogen. That's where, again, there's lots of options. Complement can come in and help kill. Macrophages, microphages can come over, cytotoxic T cells could come over, again, natural killer cells, and once the antibody is kind of attached to the pathogen, they can all potentially help kill by punching holes or by phagocytosis, okay? So when we look at, again, how our body acquires immunity, how our body defeats pathogens, all right, we have basically two broad categories of specific defenses, and it comes down to looking at the antibody production, okay? Stand by, I gotta open the door for my office mate. Okay, stand by, I'm coming back. Okay, so specific defenses, again, the antibody production here. In active immunity, we are going to say that our B cells are sensitized, our B cells are converting into a large number of plasma cells, and they are actively creating the antibodies that are being distributed throughout all of the body waters of our system. All right, so anything that's an active immunity response means we expect our B cells and our T cells, our cytotoxic T cells and our plasma B cells, but specifically for the definition here, our plasma B cells are upregulating and activating and producing antibodies that they create because they are sensitized to the pathogen. All right, passive immunity is going to be when the antibodies are not necessarily created by our B cells. So they are going to be either created in the lab, outside, by chemists, by maybe by growing them up in rabbits or mice or some other entity, harvested, collected, and then injected into us, or the more natural way, they are brought into the system because of mom, and mom shares antibodies through breast milk. All right, so when you look at these two defense systems, active is going to assume that your B cells are getting involved and making the antibodies. Passive means you're getting the antibodies out from an outside source. All right, the part about innate or naturally acquired means that you are going to, again, in some of these naturally acquired processes, you are either getting the pathogen through a natural mechanism or you're getting the antibodies through a natural mechanism. So for our four categories, again, naturally acquired passive immunity is going to be the best example. You naturally acquire a pathogen and you get antibodies in a natural way, and this is a baby, through mom's breast milk, okay? Naturally acquired active immunity is going to be you naturally, again, get exposed to a pathogen and you go through the normal processes of that pathogen ends up evading your specific and non-specific defenses for a while, and so you have to then do a better job at finding it, tagging it, and then activating your cytotoxic T cell arm, your cell mediated arm, and your B cells to make better efficient antibodies that can better tag and help you kill that pathogen. So that is like normally getting sick, okay? Now, anytime we take things into our hands outside of that natural process and we help the system, now we're going to induce artificial, either our body to make antibodies. So we go from having potentially a primary antibody response, meaning the first time it's ever exposed to a pathogen, to a secondary antibody response, which is more robust and quicker at fighting the pathogen off. Or we're going to try to either give antibodies to the system, maybe because we know our system is taking too long, our system is unable to make efficient antibodies, and we're trying to help it. All right, so artificial means something is being modulated and maneuvered outside of the body. So in our artificial passive immunity, all right, we are going to see that somebody has probably gotten sick, and we have to make the antibodies, and then we have to inject those antibodies either by drug, by shot, by IV to help the system overcome. All right, and so it could be something like the Ebola situation not too long ago. The two nurses had to get antibodies from the nurse at the CDC, or the doctor, the nurse at the CDC, that they had to harvest his blood, try to get his antibodies, and give it to the nurses so that way they could be, again, overcome the Ebola that they contracted, so they wouldn't die. It could be something like what we do with MRSA. Our system is not able to effectively kill whatever we've acquired by picking our nose and then getting a boil somewhere, and so we start giving some higher powerful IV drug antibiotics to try to help the system. You could look at it like strep. Okay, strep, when we know it's strep, we usually give penicillin or some antibiotics. Will the body potentially maybe still go through naturally acquired active immunity? Maybe, but we're not going to play the game of waiting, because either waiting might mean the person dies before we can get to that active naturally acquired immunity. So artificially acquired passive immunity is part of how we can maybe speed up and help our system overcome pathogens that are faster acting than our system's normal first time exposure primary response. Okay. On the other side, again, if we know pathogens are pretty stable and we know pathogens will probably kill us in the window of time of 3 to 5 days that it would normally take for our system to maybe even find the pathogen, get it to maybe trying to convert into specific defenses and activating antibody production, we can try again with a stable pathogen to create a situation where we expose people to the pathogen the first time in a modified, non-virulent, or dead method. And so this is where immunization comes in. So we know the flu virus mutates, so that's part of the reason why you have to get a flu vaccine every year. And what we do is we try to take 3 or 4 strands of the previous year's flu, or the flu that just happened in Australia because their winter's off of us. And what we do is then we modify it, we kill it, we don't necessarily let it be a live virus, and then we put it in a shot and we inject it into our bodies. And by injecting it into our bodies can then make new antibodies to the antigens of the flu virus that was either the year before's virus or the most recently acquired viruses from winter in the South Hemisphere. All right. And so if the flu virus in the new system that starts up this winter is close enough, our antibodies we create to that vaccine should be then sufficiently able to bind and interact with the flu virus of this year and prevent us from having, again, that long window of time of being sick, getting severely sick, because our body can mount a quicker defense. All right. Other vaccines you think about that are really stable, the polio, we've basically said we've eradicated polio. For a while, we thought we had basically reduced measles to very low incidence, whooping cough. Chickenpox is now a vaccine, and the goal is that if people still get exposed to those pathogens, they are going to have protection that is a secondary immune response, even though the first time they were exposed to a dead or modified virus. All right. Again, there's a lot of discussion on the value side of, with vaccines. There's some people we know here in St. Louis that there is Principia High School and college, and they are Christian Scientists, and they do not believe in medical interventions. So part of us all getting vaccinated is, let's say, the 1 or 2 Christian Scientist kids in our high schools or in our campus, we then potentially decrease the risk that those individuals can get measles, can get polio, could get the flu if we all got vaccinated. All right. So the reason to get vaccinated, again, this is a big point on this chapter for, again, looking at why do you have to get vaccinated if you have a job in healthcare. you have to get vaccinated because you might be working with cancer patients, with people with compromised immunities or on drugs that are suppressing their immunity. Maybe they can't take vaccines themselves. And so by you being vaccinated, the odds of then you, by herd immunity, bringing that sickness over to that compromised child or person decreases. The other thing is, it's a risk. And you see this every now and then in the news that some kids, some people die of the flu. And there is always that game of, if you don't get vaccinated and you get the virus, luck of the draw, people will die from it, because our system, again, takes time to ramp up enough and sufficiently to maybe overcome it. And so part of getting vaccinations is that for very nasty things like smallpox that left only 20% of the population able to survive getting infected and then hugely disfiguring, we changed those odds to where everybody could get maybe a little sick, but they didn't get as sick, they didn't die, and they didn't get grossly disfigured. So the other reason why we want to give vaccinations are, again, we're trying to help our bodies overcome really nasty, really deadly, really disfiguring diseases and pathogens, so that way the odds of living increase. Okay, so herd immunity and not dying from things that used to kill large parts of the population are the two big reasons why we give vaccinations. All right, for my A&P II students, expect one of these four categories: either naturally acquired passive immunity, naturally acquired active immunity, artificially acquired passive immunity, artificially acquired active immunity, to be your essay question, where you are going to, if given one of those categories, you're going to explain what it is and what it means and provide an example. And then in the case of some of them, you might need to expand and give me a good fact, so like why we want to get vaccinated versus why it might take 7 to 10 days, just some little fact about each one. Why naturally acquired passive immunity is only mother-child. So just be prepared that this is probably going to be the essay question for chapter 22. It's a big concept that you need to be very comfortable with, okay? So our next slide comes from our book, and again, it just tries to set up that for the acquired immunity, which is what we're talking about with specific defenses, that we acquire. So when you're born, you've never, been exposed to so many of the bacteria, the fungus, the virus, the everything. You have the ability to acquire immunity to those things, and you can do it in a variety of ways. Okay, innate immunity, again, part of that comes from you are human, and so hopefully swine flu doesn't attack you because you're human, because your genetics are that you shouldn't then be the pathogen's prime source. Okay. All right, so let's continue on. When we look at our immune system, there are, again, properties to how our body works with having the specific immune system the way it is. And some of those properties are listed here. So specificity, versatility, memory, and tolerance. All right. So specificity is that if I give you a certain pathogen, you're going to make a specific antigen for that pathogen. All right. That means that you will get the ability to recognize the difference between tuberculosis bacteria with an antigen versus streptococcus bacterium from the same antibody. All right, your antibody has the ability to differentiate between those two and bind to the one it's been made to bind to. All right, and so specificity is that we can create antibodies specific to certain antigens. Okay. And part of that comes from the way that B cells operate, with their ability to, when they are sensitized to an antigen. And again, this is a little complicated genetics, so we're not going to be expected to understand this in this class, but maybe in an immunology class or genetics class, they might go over this. There is some splicing and dicing of the DNA and the introns and the extrons of where the antibody is in the genome, and B cells forever then create an antibody that they will then always make that's specific to an antigen. Okay. Now, versatility of the system is that right now, the next breath you breathe, let's say there is some fungus in there, let's say there is viruses in the air, there's bacteria in the air, and oh, let's just throw in, you sucked in a few protists, all right? The versatility is that you can handle all 4 different types of pathogens. You can handle multiple pathogen, different viruses, different bacteria that you take in a single breath, a single bite of food, and your system can handle that there is a lot of different pathogens from a lot of different sources coming in at a lot of different means, a lot of different ways, and you have the ability to still fight that. Chances are you are probably actively fighting 2 or 3 things from the mouth, from the nose, and then a different 2 or 3 things in your urethra, around your anus, and then in your lungs. Okay. Memory comes from the way the system sets up that T cells and B cells, after they've been sensitized, they will, again, mostly mitosis into active living cells for the moment, but some of those cells will become pushed into memory cells. And our memory cells can live, as we talked about earlier, somewhere around 2-5 years, maybe even as long as 20. And so we then get that lifetime immunity. So that's part of the fun of, if you have had chickenpox or the chickenpox vaccine, you should never have chickenpox for a second time. The same thing with the smallpox. Once they gave the vaccine, the odds were you would never have smallpox. You would have that lifetime immunity because of those memory cells. All right, and those memory cells will, when exposed to the antigen and subsequent time frames and manners, can quickly, again, mitosis, go through and ramp up into more plasma cells, more antibodies. Your titers, your levels of antibodies in circulation can upregulate, and you can create new memory cells for subsequent and follow-on life. Okay. And then the last part of this whole thing is that we have tolerance. So in theory, the antigens you're targeting are always with immunopotent T cells, non-self antigens, and so your system shouldn't attack your body. All right. Now, with this system, if you get a organ transplant from a different person, that is part of what we then have to modulate that tolerance, because now you potentially have a kidney or a heart or lungs that are technically not your genetic cells. And so some of the proteins, glycoproteins, and different antigens on those cells of that organ might then be not self. And so that's why certain people, depending upon the organ, depending upon how closely it is to them, might have to be on immune suppressant drugs and protocols for the rest of their life to ensure that they then do not destroy the organ that they received from whatever reason that they've received the organ. Okay. All right, moving onward. Okay, so here's kind of, again, our overall. Once you activate your immune response, we expect that our T cells, our cytotoxic T cells, our killing of the cells out there expressing the antigen, killing of cells, actively hijacked, expressing the products of a pathogen that's ingested or gotten in there, will be activated, along with B cells to make antibodies that can go through all of our body waters. So they can try to find all the little nooks and crannies of connective tissue, all the little spaces and places that, again, pathogens might try to, hide. All the little closets that pathogens might try to hide into. Okay? All right. So the big question is, how do we transition to these 2 acquired active processes? All right. And what we learned with HIV is that there are T helper cells that are going to be the critical link. And the reason why we assume that it's T helper cells here is because in HIV patients, as the virus which attacked their T helper cells killed off the T helper cells, all of a sudden they were not able to ramp up antibodies, and they started having lots of sickness and death associated with pneumonia, with other normal pathogens that, again, our body should have been able to defend against if we had those T helper cells available. Okay, so transitioning to active cell-mediated immunity, active antibody humoral immunity seems to be with respect for what we know in the book. And again, I do want to specify that our book might be out of date, might be wrong, might be not totally current on the immunology research, but we think it's the T helper cells. Okay, so if T helper cells randomly come into contact with pathogens, part of what they will do is recognize that the pathogen is not self. They will then send some of their cytokines out. Remember the interferons, interleukins, colony stimulating factors, a bunch of those proteins, and it will call over some of our, again, T cells, B cells, and the T cells, the cytotoxic T cells, can then be introduced to the antigen that is now not self and sensitized to, okay, now go and find this antigen everywhere it's displayed and kill those cells displaying it. Okay, so T cells have to, again, be immunocompetent. They have to recognize antigens. They might be doing that just randomly because pathogens run into them in all the places the T cells live. The other way to bring antigens to the T cells are through those antigen presenting cells. And this is where macrophages, neutrophils, eosinophils, anything that ingests pieces and parts of cells or whole cells like macrophages do, they digest those materials, they take pieces and parts of those materials, and they can display it on their membrane, bring it over to the T cell, and I kind of think of it as, hey, look what I ate. And the T cell looks at what they ate and says, oh, that's just some debris from broken cells that we need to clean up, or, hey, that is something not self. Same thing, once the antigen presenting cell and the T cell decide what the antigen presenting cell ate is not self, the T cell will release cytokines, draw in more cytotoxic T cells, more B cells, and will kind of like turn on the forces of get active, find cells displaying this pathogen and kill them, get active, make lots of antibodies so we can find all the places this antigen is hiding. Right? So transitioning to both cell-mediated and humoral immunity seems to be related to, again, from our HIV work, something related to a T helper cell. The T helper cell either finds the antigen, just blind luck, runs into it, or gets presented the antigen by a phagocytic cell, which we call antigen-presenting cells. Okay, and that can happen anywhere that T cells and antigen-presenting cells are located. All right. So part of how we know the T cells recognize antigens is that we know that our cells genetically are going to make a protein on our membranes that's called a major histocompatibility complex. And I put this picture here up from a while back of on your membrane. Remember, you have lots and lots of proteins, glycoproteins, phospholipids, glycolipids. You have lots of things put on there. And part of what your T cell does is it knows some of these molecules if they belong or don't belong. Somehow it knows your genetic code and what your genetic code should produce. And so the major histocompatibility complex, our MHC, is genetically determined. And it's one of these proteins that ends up on our membrane of all of our cells, and then another type ends up on the membrane of all of our antigen presenting cells. So one of the ways your T cell knows every cell is either self or not self is it runs into this MHC molecule, and if the MHC molecule is correct because the genetics that created it is your genetic code, then yay, the T cell knows yourself. All right. The other thing the major histocompatibility complex gives us is it has a pore. And in that pore, what we can do is either put some of the protein, some of the products, some of the molecules from inside of the cell, and we can display them on the outside. So this is how not only can T cells go up to a cell and say, hey, are you self? It can also go up to a T cell and say, hey, what are you making? Or what are you producing in large amounts through your protein synthesis or through your other synthesizing processes? And so if, let's say, the cell has been hijacked by a virus and it's producing all these viral antigen proteins and toxins and DNA, RNA particles, what your major histocompatibility complex MHC molecule can display is some of those products that it's making inside, and the T cell can go, hey, you're self, but what you're making is not self. So that's kind of the gist of part of how T cells might be able to, one, tell what is self versus not self, and then for things that are self, maybe if they are making inappropriate or abnormal or non-self pathogen hijacked material. All right. Now, for the antigen presenting cells, they also have an MHC complex, and their MHC complex can do just like the other one, the normal, every cell in the body, take some of what we're making, put it on there, but they have a different MHC complex that also lets them, when they phagocytose molecules, pieces and parts of cells, or full cells, they can break down those full cells, take pieces and parts of what they ate, and display it. And so that's how they take a part of the pathogen that they ingested and show it to the T cell, because again, the pathogen doesn't have the genetic code of your genes. That antigen should be something that is not self. And so the cell is, yes, self, because it's your macrophage, my neutrophil, but what the neutrophil ate is therefore shown to be not self. All right. So we have to have then the T cell and the cells of the body, and the T cell and the antigen presenting cells have to have these corresponding little proteins that want to hook up and check on each other. All right. So that is where the end of the chapter comes in. And again, we have two types of MHC molecules. All right. MHC1 would be the MHC molecule maybe on all of the cells of your body, and it's how all of the cells of your body interact with T cells, and the T cell knows yourself. But it's also how the T cell interacts with the, again, cells of the body and sees what are you doing in there? What are you making? What's quality control? What's happening in here? And then those molecules that are being made, if they are, again, parts of a pathogen, products from a pathogen that we have found to be in our system and be not self, the cytotoxic T cell can then step up, recognize you're a cell that belongs to our body, but you're a cell that's been hijacked, so let's kill you. All right, so that is a class 1 HC molecule, and that should interact again with your proteins, molecule receptor protein specific to cytotoxic T cells. Okay, now again, the MHC class 2 molecule, which is specific to our phagocytic antigen presenting cells, this type of MHC we know is going to interact with some of our T helper cells. And it's going to happen to some of our T helper cells that have a different little receptor protein that binds to it. Again, T helper cell, by bringing these molecules together, first says, hey, macrophage or neutrophil or antigen presenting cell, you are self. But then it gets to look at, hey, what did you eat? And if what you ate is not self, it can then target that for, okay, let's bring everybody over here. Let's get everybody aware that this is now in our body. Let's find it, and then let's go ahead and kill it, and kill maybe you as well. All right. So again, we have two types of MHC molecules. MHC class 2 is associated with our antigen presenting cells, our phagocytic cells. MHC class 1 would be potentially on every cell of the body. These are part of maybe how T cells know immunocompetence, if a cell is self or not. And then it's how a T cell gets some insight to what is going on inside a cell, or maybe what did a cell eat. All right. Now, again, there's a lot of kind of holes in this chapter, but for what you need to know, I want you to get comfortable with MHC class 1 proteins on all the cells of our body are going to be matched up and kind of linked to any immune cell, T cell, that displays what we call a CD8 protein. All right, so the CD8 protein is what is binding to the MHC molecule. So those two entities, kind of in that previous picture, come together. And that's part of how the cell knows, okay, yourself, and then the cell gets in a docking situation where the T cell potentially can then gain some insight and look at the protein product displayed in the MHC1 area and determine if the cell needs to be killed because it's hijacked or not. Okay, so CD8 T cells are associated with cytotoxic T cells, memory T cells, and suppressor cells. So this is how, again, if chickenpox is in your system, the cytotoxic T cells are circulating and finding all of the cells in our body that are currently producing chickenpox virus, proteins, particles, toxins, whatever, and killing them. All right, but all of the cells may be infected or not actively displaying those items. So we make some memory cells, and those memory cells later on, as maybe rogue and intermittently cells start making antibody or making antigens related to the chickenpox virus because they were previously infective, the memory T cells would be able to restart the system and kill off those, again, chickenpox virus producing cells. And they do it so quickly that you never then have another chickenpox outbreak. All right, so that's kind of how that works. All right, so the takeaway is CD8 and MHC1 go together. All right, now phagocytic cells have potentially MHC1 molecules, but they also have that MHC2 molecule. And that MHC2 molecule lets them, when they phagocytose material, take part of that piece material, toxin, display it, they are going to interact with CD4 T cells. All right, and CD4 T cells can be some of our helper cells. Okay, and they then are going to be the CD4 protein and the MHC2 protein hook up. That's how, all right, yourself. And what did you eat? Oh, you ate something that's not self. Let's go ahead and try to kill it and kill you. Okay, and bring over more B cells and more T cells to then also identify and get ramped up to find this antigen as well. All right, so the takeaway is MHC class 2, CD4, MHC class 1, CD8. All right, and some of the specifics of all of this, again, the book is not very clear. If you go on the internet, there's lots of different theories out there, or we haven't quite narrowed it down. So I don't need you to tell me for sure CD4 is always a helper T cell. It could be in some situations, maybe the T cell that has CD4 could still become a cytotoxic. So I don't want to get too caught up in all of those details, all right? For now, cytotoxic T cells actively trying to kill cell-mediated active immunity should have CD8 and should be looking for the antigen and the MHC1 molecules. Helper T cells sitting in your lymphocytes, sitting in your spleen, waiting to get activated, waiting to initiate a new cell-mediated and humoral response are going to be some CD4 protein expressing T cells, and antigen presenting cells will interact with them when they eat things and show them what they eat. And if what they eat happens to be from a pathogen, the CD4 can activate and ramp up new responses to then find and target those antibodies. That's our takeaway. Okay. All right, for our B cells. Okay, and again, some of the pictures here show that B cells have a class 2 MHC, which would say, hey, they are an antigen presenting cell. Again, we are not going to focus on all of those details. What we are going to focus on is that naive B cells will tend to be called over to a new antigen finding by a T helper cell and will then get in contact with that antigen, and we believe turn and do some different genetic splicing and dicing, and will then become plasma cells making large amounts of antibody that's now specific to the antigen the T helper cell introduced them to. And so when that happens, there will be lots of plasma cells making lots of protein products, antibodies, and the antibodies will release. And then every now and then, one of these mitosis reactions will lead to a memory cell. Okay, so that's what I want you to take away with the B cell. All right, again, that antibody, the genetic code has some parts to it that are always the same, the constant region. And then if you go back to the complement, remember the C1 protein and complement molecules binds to the heavy chain, the constant region. So we want that constant part and that complement to always be able to find each other. So that would be why we don't want to mess with that genetic code. But what we do want to mess with on the light chain is this variable region. So if the antigen maybe has lots of hydrophobic amino acids, this would be the region where we try to create some hydrophobic amino acids that'll then bind and more likely hook up into with the antigen. Okay. Again, I don't need you to know the specifics of how we splice and dice. You do need to know that these proteins tend to be drawn as Ys, and we call them immunoglobulins. So Ig, capital I, little g, and then again, some of the specifics of which intron and extron and what kind of pathogens the protein ends up being made to find might specify if it's IgA, IgM, IgG, but I am not going to go into that type of questioning since this is not an immunology class, not a microbiology class. Just for me, know that these are antibodies. The variable region is what you mix and match to then always find your antigen. Okay, and so again, humoral immunity, it's not about killing the pathogen, it's about finding it. And the finding it comes from making lots and lots and lots of proteins that have an easier ability to spread through all the body waters. We keep that kind of protein coding always and forever with some of our memory cells that can then, upon interaction with antigens in the future, reactivate and form new plasma cells and recreate more antibodies and ramp up our titers. All right. So as we start to look at, again, our specific defenses and our non-specific defenses together, you see that the activity from the cells are going to somewhat fall into what cells are more likely to be involved in non-specific defenses and then ones that are going to be more involved in the specific defenses. And you can see the timing is macrophages, natural killer cells, neutrophils, antigen-presenting cells that are all part of that. Again, phagocytosis, immunosurveillance, part of our non-specific defenses tend to have more activity if a pathogen enters our system in the early time points, whereas our cytotoxic T cells get active, our plasma cells, and then our antibody levels increase more towards the back end after we've had time to try to potentially find the pathogen, sensitize to it, and realize where it's located, find it, and kill it. Okay? And so what we look at for a primary response in the first time we activate plasma cells, get antibodies to, again, increase in number, is we have what's known as primary response. And mostly, again, don't worry about the IgM versus IgG. Just know that for the first time you come into contact with an antigen, again, it might be very good at evading, at not getting caught, not getting phagocytosed. And so it might be that the pathogen is propagating and spreading and having its field day before we then, maybe 72, maybe longer, finally maybe find it, have T cells interact with it and activate cytotoxic T cells and B cells. And so it might then be 1 week before you start to have any type of significant amount of antibody in circulation that is able to, specificity-wise, bind and help tag that pathogen. Again, for certain diseases, if I give myself 5 to 7 days, that disease will kill me in 3. So primary antigen response are not necessarily ideal with certain diseases. So why vaccinations are so important is that it allows us potentially to, again, introduce a pathogen, but it's a pathogen we've seen before. So there's memory B cells in place. And if those memory B cells are still alive and kicking and able to interact with the pathogen and the antigen of the pathogens, they can very quickly, again, because of that specificity, recognize, hey, this is something we're geared to fight, ramp up more plasma cell numbers, more antibody numbers, and very quickly within hours to a day or two, get lots and lots of antibodies circulating, finding maybe the pathogen before it has as much of an opportunity or as much of a timeline to undergo its own mitosis and propagation. And so that's part of the reason why you might not ever get sick, or like with the flu virus, you always hear people, I took the flu vaccine and I still got the flu. Well, trust me, you got the flu that was maybe 3 days of being sick, not the flu of a week to 10 days in the hospital almost dead. So it's not perfect, but it will significantly potentially reduce how sick you get and reduce the risk of dying from that sickness. All right. So it all comes together again, our system here with antigens get into our system. Antigens are hopefully found quickly. If they are not, it could be that you are spreading and propagating and even contagious and keep spreading it to other people as well as to your system. But hopefully our nonspecific defenses do the decent job of locating, finding, and showing it to our T cells and getting our activation of our cell killing side and our tagging side of our system quickly up and running before we either die or disfigure or have really terrible days, weeks of pain, fever, and discomfort. Okay, so that is, again, our chapter. Hopefully this explains a lot of the back end. There are a lot more details in the book. And again, the book has still got some big holes in it because our book is not an immunology book and it's not written by immunologists. And remember, the field of immunology, the field of this whole process is still rapidly changing and getting more understood. And so our books are 20 years behind. So if in the event you become part of an immunology or public health, or you take some classes later in life, they will probably correct all the wrong things that I have probably just lectured you on. But until then, this is what we're going with for now. This is what you need to learn for now, for 2019, for your test coming up with Dr. Granier. All right, I'm going to stop the recording, and I'll get, hopefully, any other questions, please feel free to email me or message me. I guess I could talk, Bana. I'm like typing all of this, but I guess I could talk to you. So, so if the B cell is immunocompetent, that's the big question. Yes, a naive B cell interacting with a pathogen or an antigen could activate, all right, and become, start humoral immunity. But part of the early part of the chapter and part of the early data shows that T cells become immunocompetent by going to the thymus gland. B cells are not immunocompetent and they stay in the bone marrow. So again, the theory would be somehow the B cells have to be immunocompetent. All right. So the best way for naive B cells to activate is cytokines are released from um, your T helper cells that are in contact with an antigen presenting cell and or a T helper cell that's in contact with a pathogen. The T cell is immunocompetent. The T cell knows that the antigen is not self. So then the T cell says, hey, B cell, come over here, and introduces them. All right. So when I took my all of my immune courses, and this was back in 2005, 2006, somewhere in there. The instructor led us to believe that, again, the best way to get a B cell activated that is not been exposed to any pathogen, period, end of story,